Damage caused by ultraviolet radiation. Ultraviolet radiation cross-links adjacent pyrimidines on the same DNA strand, forming two major lesions. 80 to 90% of these are pyrimidine dimers, which are also called cyclobutane pyrimidine dimer, because of the four-member cyclobutane ring that forms between the two bases. Pyrimidine dimers, which are also called cyclobutane pyrimidine dimers or CPDs, because of the four-member cyclobutane ring that forms between the two bases. 10 to 20% of the lesions are photoproducts in which the 6 carbon of one pyrimidine is linked to 4 carbon of adjacent pyrimidine. Both of these products block DNA replication because they are non-informative, non-coding. The replication machinery cannot tell which bases to insert opposite the lesion. As well as we will see, replication sometimes proceeds anyway, and the bases are inserted without the benefit of the base pairing that normally provides accuracy. If these are the wrong bases, a mutation results. Then damage caused by gamma and X-rays. The much more energetic gamma rays and X-rays, unlike UV rays, can interact directly with the DNA molecule. However, they cause most of the damage by ionizing the molecule, especially water surrounding the DNA. This forms free radical chemical substances with an unpaired electron. These free radicals, especially those containing oxygen, are extremely reactive and they immediately attack neighboring molecules. When such a free radical attacks a DNA molecule, it can change a base or it can cause a single or a double stranded break. DNA bases are subject to at least 20 kinds of oxidative damage, and these can be caused by reactive oxygen species derived from ionizing radiation or simply from normal oxidative metabolism. The best studied oxidative damage of DNA bases 8-oxoguanine, also known as 8-hydroxyguanine. DNA polymerase in bacteria and eukaryotes mislead OXOG as thymine and insert adenine instead of cytosine, resulting in an OXO-GA pair. Both bases in this pair are genotoxic because they both will probably lead to mutations if they are not removed before the DNA replication again. Single-stranded breaks are ordinarily not serious because they are easily repaired just by rejoining the ends of severed strand, but double-stranded breaks are very difficult to repair properly, so they frequently cause a lasting mutation. Because ionizing radiation can break chromosomes, it is referred to not only as a mutagen or mutagen causing substance but also as a clastogen, which means a breaker. Next, repair mechanisms of DNA. DNA repair mechanisms act to prevent mutation and necrobiosis, thus maintaining genome stability, cellular integrity, and preventing the onset of cancer, degeneration, and premature aging. First, direct reversal. Photoreactivation repair refers to DNA repair system where the re-exposure repairs the UV damaged DNA under the visible light. It is also called the direct reversal DNA repair mechanism, in which the photolyse enzyme under the action of the visible light reverses the effect of UV radiation on the cell. Then photoreactivation repair. DNA with lesion pyrimidine dimer, photo reactivation by visible light exposure to UV radiation, normal DNA without dimer. UV exposure, UV light, dimer formation created. Mechanism of photoreactivation. The photoreactivation process repairs the UV damaged DNA. A DNA helix becomes distorted and forms a pyrimidine dimer, usually thymine-thymine dimer, under UV light exposure. A dimer forms when the adjacent thymine bases attach covalently to their respective C5 and C6 forms. The formation of TT dimer causes helix distortion, which eventually causes replication errors in the DNA and finally leads to mutation. The covalent bond formed between the two adjacent thymine bases must be broken to recover the helix distortion. An enzyme photolyase breaks off the covalent bond between the dimer. Then formation of pyrimidine dimer. UV light induces a covalent bond between the pyrimidine bases, either thymine or cytosine. The two adjacent pyrimidine bases bind through a carbon-carbon double bond. A carbon-carbon double bond formation between the pyrimidine bases form a dimeric structure commonly referred to as cyclobutane pyrimidine dimers. Cyclobutane pyrimidine dimers, CBPBs, is a common product forming under the UV exposure. In addition to this, 6 to 4 photoproducts also produced as a result of UV damage, where the two bases attach with a single carbon bond to the C6 of one ring and C4 of another ring. thymine dimer usually forms after the UV exposure and it appears like a bubble in the DNA strand. Treatment with photolyase under visible light. Under the visible light of a longer wavelength, a photolyase enzyme becomes activated. A photolyase enzyme scans the DNA strand and recognizes the pyrimidine dimers. After recognition of dimeric forms in the DNA, it directly binds to that segment. Then breaking of TT dimer. When the photolyase enzyme binds to the TT dimer, it starts splitting the carbon-carbon double bond, covalent bond between cyclobutane rings. Thus, a photolyase enzyme converts the dimeric form of pyrimidine bases into monomeric forms. Photoreactivation repair, DNA strand, TT dimer, covalent bond. Act directly on damaged nucleotides, converting each back to its original structure. Only some kinds of damaged nucleotides can be repaired directly. X can be repaired only by a DNA ligase if all that has happened is that a phosphodiester bond has been broken without damage to the 5-phosphate and 3-hydroxyl groups of nucleotides either side of a nick. This is often the case with nicks resulting from the effects of ionizing radiation. And alkylation damage. Some forms of alkylation damage can be directly reversed by enzymes. In mammals, MGMT or AGT repairs two varieties of DNA adducts, O6-methyl guanine and O4-methyl thymine. So removal of O6-MEG and O4-MET modifications are achieved by a one-step methyl transferase reaction. MGMT accepts the alkyl adduct from the modified oxygen molecule, directly restoring the DNA base and inactivating itself. Cyclobutyl dimers are repaired by light-dependent direct system called photoreactivation. Then mismatch repair. DNA mismatch repair or MMR is a system of recognizing and repairing erroneous insertion, deletion, and misincorporation of bases that can arise during DNA replication and recombination, as well as repairing some forms of DNA damage. Mismatch repair is strand specific. During the DNA synthesis, the newly synthesized daughter strand will commonly include errors. In order to repair begin, the mismatch repair machinery distinguishes the newly synthesized strand from the template or parental strand. And examples of mismatched bases include a GT or AC pairing, cDNA repair, when mismatches are commonly due to tautomerization of bases during DNA replication. A DNA mismatch generates due to the incorporation of an incorrect base during the DNA replication process. The mismatch repair system uses the methylation on the parental strand as a signal to leave that strand alone that correct the nearby mismatch in the unmethylated progeny strand. Mut S alpha recognizes base-base mismatches and mut L alpha nicks the 3' or 5' side of the mismatched base. The nicked DNA segment is then excised by the XO1 exonuclease in corporation with the single-stranded DNA binding protein RPA. So DNA polymerase delta and DNA ligase synthesizes the DNA strand. Mismatch repair depends on a number of activities in human cells, including mut S alpha or mut S beta, mut L alpha, the exonuclease XO1, DNA polymerase delta, the replication clamp PCNA, the clamp loader RFC, replication factor C, the single-stranded DNA binding protein RPA, replication protein A, and the non-histone chromosome protein. The first step in mismatch repair is recognition of the error. The method of strand discrimination, which strand is the daughter strand, with the mistake in mammalian cells is unknown. One model proposes that the 5' end of the Okazaki fragment and the polymerase machinery associated with the 3' end of the nascent strand may provide markers for strand discrimination in post-replication mismatch repair. XO1 in association with PCNA then progressively removes the portion of the strand in between the nick and the mismatch. HMGB1 plays a role in stimulating XO1 activity. Next, repair synthesis to replace the excised strand with new DNA is mediated by DNA polymerase delta and its associated factors. Finally, DNA ligase 1 seals the nick by forming the last phosphodiester bond. General steps of DNA mismatch repair: mismatch recognition, recruitment of related proteins, excision and replacement, and gap filling. Mismatch recognition in prokaryotic MMR. The MUTS protein complex detects the DNA lesion and combines with the ATP molecule and gets activated. In eukaryotic MMR, the MSH protein complex recognizes the non-matched base in the DNA strand and gets activated by an ATP molecule. Then recruitment of related proteins. In prokaryotic MMR, the activated ATP MUTS complex recruits MUTL, acts as a carrier protein. This MUTS and MUTL complex activates the MUTH complex, MUTS, MUTL, and MUTH from the tertiary complex, which loads the DNA helicase 2 or UVRD enzyme onto the DNA lesion site. In eukaryotic MMR, the activated ATP MSH complex recruits pCNA and polymerase delta. Then ATP-dependent conformation change occurs in the mobile clamp. The conformational changes recruit the binding of the MUT-LA complex, the pCNA, profiling cell nuclear antigens. In prokaryotic MMR, the activated ATP MUTS complex recruits MUT-L, acts as a carrier protein. Then MUTS and MUT-L complex activates the MUT-H complex. MUTS, MUT-L, and MUT-H form a ternary complex which loads the DNA helicase 2 or UVRD enzyme onto the DNA lesion site. In eukaryotic MMR, the activated ATP MSH complex recruits pCNA and polymerase delta. Then ATP-dependent conformation change occurs in the mobile clamp. The conformational change recruits the binding of the MUT-LA complex. The pCNA proliferating cell nuclear antigens activate the MUT-LA complex and create a nick in the daughter strand. Excision and replacement. In prokaryotic MMR, exonuclease 1 is activated by the MUT-SA protein, which removes the mispaired bases. Then RDA protein displaces the mismatched base. MUT-SA and MUT-LA terminate the exonuclease 1 activity. In eukaryotic MMR, the MUT-LA complex promotes the digestion of the mismatched bases by the exonuclease activity 1 and association of RPA, recognition replication protein A. Gap filling. In prokaryotic MMR, the DNA polymerase 3, though through its 5' 3' activity, forms new bases. Single-stranded binding protein SSB also bound to the new DNA strand and prevents the new DNA to become double-stranded. Finally, DNA ligase fills the gap in the DNA strand. In eukaryotic MMR, the RFC, replication factor C enzyme, plays an important role in the synthesis of new DNA bases. Replication factor C enzyme attaches at the 3' primer site and activates the DNA polymerase activity to form new bases. Single-stranded binding proteins prevents the new DNA to become double-stranded. Finally, DNA ligase binds the gap in the DNA strand. Then excision repair. Nucleotide excision repair. The nucleotide excision repair pathway is used for repair of structural distortion. For example, bulges from thymine dimers induced by UV irradiation. In humans, nucleotide excision repair is carried out by six repair factors: RPA, XPA, XPC, TF2H, like that. So ERCC1 stands for excision repair cross complementing rodent repair deficiency. Then bulky base damage, including pyrimidine dimers, can be removed directly without help of a DNA glycosylase. In this nucleotide excision repair or NER pathway, the incising enzyme system recognizes the strand with the bulky damage and makes cuts on either side of the damage, removing an oligonucleotide with the damage. The key enzyme E. coli cells use in its process is called UVR ABC exonuclease because it contains 3 polypeptides, the products of UVRA, UVRB, and UVRC genes. This enzyme cuts the damaged DNA, producing an oligonucleotide that is 12 to 13 bases long, depending on whether the damage affects one nucleotide or more. Next, base excision repair, recombination repair, some other factors of SOS response, mechanisms of SOS repair, etc. are present. So next one is excision repair, in the sense nucleotide excision repair. The nucleotide excision repair pathway is used for repair of structural distortion. For example, bulges from thymine dimers induced by UV radiation. We just learned this. Then next is base excision repair. In base excision repair, a damaged base is recognized by an enzyme called DNA glycosylase, which distors DNA in such a way as to extrude the damaged base out of its association with its base pairing partner, then breaks the glycosidic bond between the damaged base and its sugar. This leaves an apurinic or apyrimidinic site, which is a sugar without its purine or pyrimidine base. Once the AP site is created, it is recognized by an AP endonuclease that cuts nicks and DNA strand in the 59 side of the AP site. The endonuclease means the enzyme cuts inside the DNA strand, not at free end. Greek endo meaning within. Base excision repair typically acts on subtle base damage. This process begins with a DNA glycosylase which extrudes a base in a damaged base pair, then clips out the damaged base, leaving an apurinic or apyrimidinic site that attracts the DNA repair enzymes that remove the remaining deoxyribose phosphate and replace it in a normal nucleotide. In bacteria, DNA polymerase 1 is an enzyme that fills in the missing nucleotide in BER. In eukaryotes, DNA polymerase B plays this role. However, this enzyme makes mistakes and has no proofreading activity. So APE1 carries out the necessary proofreading. Repair of 8 oxoguanine sites in DNA is a special case of BER that can happen in two ways. Since OXOG mispairs with A and A can be removed after DNA replication by a specialized adenine DNA glycosylase. However, if replication has not yet occurred, the OXOG will still be paired with C and the OXOG can be removed by another DNA glycosylase and the OXOG repair system. Next, recombination repair. Recombination DNA repair is a biological comparative process in response to DNA damage caused by exogenous agent like UV light. The process is done by generating functional DNA by breaking and rejoining DNA strands and repairing based on homologous unaltered DNA strands. The cell has repair mechanisms to correct DNA damage. For instance, a cell that is exposed to high energy radiation could cause the chromosome to break into two pieces. The cells repair the broken chromosome by non-homologous and end joining or by homologous recombination. The protein with double-stranded breaks or DSBs is the possibility of losing a large portion in the chromosome at the breaking point. To repair a non-homologous repair mechanism, the broken ends of the chromosomes are joined back together. Since a portion of the chromosome might have been lost following breakage, the mere end joining of two chromosome segments at the breaking site could likely lead to mutation due to lost nucleotides. This is somehow avoided in the repair strategy is by more complex homologous recombination. In the homologous recombination repair process, the nucleotide sequence from the other parts of homologous chromosomes called homologue is an example of the template used to create the lost portion of the damaged chromosome. Homologous chromosomes are paired chromosomes in the nucleus. In the homologous recombination repair process, the nucleotide sequence from the other pair of homologous chromosomes called homologue is an example of the template used to create a lost portion of the damaged chromosome. Homologous chromosomes are paired chromosomes in the nucleus. Chromosomes that have the same gene loci, gene sequence, centromere location, chromosomal length, but may have different alleles are referred to as homologues. Humans in particular have 23 pairs of chromosomes, 22 of which are homologous autosomal. Half of them are paternal, whereas the other half are maternal. These chromosomes pair up during meiosis, but not during mitosis. Recombination repair is a mechanism for generating a functional DNA molecule from two damaged molecules. It is an essential repair process for dividing cells because a replication fork may arise at a damaged site, such as a thymine dimer, before the excision repair system has eliminated damage. Then SOS response. The SOS response was discovered and named after Miroslav Radman in 1975. The SOS response is a global response to DNA damage in which the cell cycle is arrested and DNA repair and mutagenesis is induced. The system involves the REC-A protein. The REC-A protein stimulated by single-stranded RNA DNA is involved in the inactivation of the repressor LXA of SOS response genes, thereby inducing the response. It is an error-prone repair system that contributes significantly to DNA changes observed in a wide range of species. SOS repair system damaged, then SOS repair refers to the DNA repair system which uses REC-A. SOS repair refers to the DNA repair system which uses REC-A regulatory protein to inhibit the repressor's activity and activate the SOS inducer genes to recover the DNA damage. It stands for Save Our Soul. The SOS system remains repressed until the conversion of REC-A protein into REC-A protease. It does not repair the DNA damage completely but provides tolerability to the affected organism. SOS system only activates in case of excessive DNA damage leading to single-strand breakage at the replication fork. This DNA damage activates the REC-A regulatory protein that links with the single-stranded DNA without the cellular energy ATP. The mechanism of SOS repair. The mechanism of SOS repair is a complex cellular process mediated by the organism itself, include the following steps. In case of excessive DNA damage, stress conditions, etc., cell responds by activating signal or REC-A protein. It floats in the vicinity of the cell in search for any damage in the DNA. The REC-A protein significantly binds to the single-stranded DNA. On binding with single-stranded DNA fragments, REC-A forms a filament-like structure around the DNA. When a LEX-A repressor comes in contact with the nucleoprotein filament assembled by the REC-A protein and REC-A interacts with the repressor protein, it converts into REC-A protease. The formation of REC-A protease causes autocatalytic proteolysis of LEX-A repressor protein. Thus, a LEX-A protein could not bind with the SOS operator. Then an activation of LEX-A protein activates the inducer protein that repairs the DNA damage but alters the DNA sequence. After DNA repair, the REC-A protein loses its efficiency to cause proteolysis, and the REC-A protein will again bind to the SOS operator or switch off the SOS system. REC-A protein, REC-A protease, damaged DNA, LEX-A protein, LEX-A inactivation, LEX-A repression, LEX-A protein, autocatalysis, repression, the promoter of LEX-A, expression, promoter of LEX-A.